Buongiorno. How you doing? Let us continue our study of anatomy and anatomical skills by making you experts on microscopy, which is going to be a little difficult without your hands on a microscope. You might open up your lab manual and look for the diagram of a microscope, and I'm going to go over that. Then I'm going to talk about how to calculate what magnification the microscope is set for and what that means. And then I am going to talk to you about some general microscopic principles, not microscopic principles, principles of microscopy. See how that's different? Microscopic principles would be very small ones indeed. Principles of microscopy, that would be how it works. So you get your lab manual and get it open to the microscopy page. I'll wait here while you do that. Okay, got it. Done? All right, good. What do we have here? Look at this. This is a picture through a microscope. A picture through a microscope is called a micrograph. And we have one here. If we want to study cells, and as I've said before many times already, physiology happens at the cellular level. And we're going to look at some of the processes that take place in cells. A group of cells cooperating to perform some function is a tissue. And after our lab exam, I will have a section on tissues. And so in order to see the cells that make up tissues, we've got to use a microscope. And so we need to learn about microscopy, how it all works. The cell undergoing cell division, a cell division known as anaphase of mitosis. You can see that right there. The AN in this case doesn't stand for absent or without. It's part of the prefix ana, which is not on your list, but that means movement. And the chromosomes have separated and are moving to opposite ends of the cell. We will get to mitosis in the not too distant future. So this is a compound microscope. It gets its name because there's two sets of lenses. The first sets of lenses are the ones that you look through. Some people might call them an eyepiece, but they're called the ocular lenses. And there's one or two. There's not usually three, because people only have two. Why would there be three? Actually, there are some microscopes for teaching purposes that have two sets of oculars. They have four. So one person watches while the other one manipulates. So ocular lens. So what do we want to do with this information I'm giving you on the exam? There's going to be a diagram of a picture like this and a pointer saying, name this microscope part. If it's pointing at the here, you would call it the ocular lenses. See, look at that. Lens is singular. Lenses is plural. Oh, this is a little bit wrong here because it says lens, which means singular, but it's got two arrows. All right. What is this? This is the diopter adjustment. That's not on your list of things you got to know. But what it is sometimes people, their eyes are different. One eye is different than the other, so the lenses are adjustable so you can compensate for differential vision. We take a look here. This is called the arm, and down here where it sits is the base. If you are going to actually pick up a microscope and take it somewhere, like take it out of the storage area and put it on your lab bench, you would hold it by the arm and support it by the base, and be very gentle with this because it's a fine instrument. They're very expensive, and if you bang them around, everything gets loose and out of focus, and well, that's just not good, and we need to be nice to them. Another set of lenses we have here are the objective lenses. Oh, there we go again, singular with two arrows. And the objective lens gets its name because it's closer to the object that you're looking at. On this one, we have a rotating nose piece that the individual lenses are screwed into, and they are lenses of different magnifications. And the way you would switch from one to another is you would just turn it, and it would click into place. And the one that you would be observing with would be this one pointing right down here at this flat area, which is called the stage. Now, when you go to use a microscope in real life, always, always, always use the low power lens first. The way microscopes work is that they're really easy to use at low power. It's easy to find your object, it's easy to get it in focus. It's just not as high magnification, and the higher magnification you use, the more difficult the instrument is to use. It's more difficult to focus, it's more difficult to find your object, it's more difficult to everything. At low power, there's just no other way to make it work. It just doesn't work. Once you get even reasonably proficient with a microscope, it'll be nothing. You'll start it, it'll be like instinct. You'll just start at low power, you're looking at boom. It'll only take you about 10 seconds to find your object and get it in focus. And yeah, I wonder what that is. I hope it's just allergies. And then you make sure your object is in the very center of view, and then you switch lenses. So we go back to this picture, and we see that the chromosomes are right in the middle of the picture. So if we want to, now this is a high magnification. It doesn't tell us what the magnification is, but this is 1,000 times or better. So there's really not a whole lot higher magnification than this, but that there is. And we wanted to focus in on maybe look at this area. Well, what we would have to do is we would have to move that over right into the middle of our field of view, because we're going to zoom in on it. And let's say we want to look at something over here, but when we zoom in, we're only seeing this much space. We won't see anything. So we get our object in focus at low power, and then we find exactly what we want to look at, and we put it right in the middle of our field of view, and then we switch to the next power, the medium power. Now, the microscope has two adjustments. And where's the other one? All right, this is a little different than the microscopes we have. Ours have two knobs. It has a big knob on the outside called the coarse adjustment, and a smaller knob inside called the fine adjustment. And the coarse adjustment, if we were in lab, I'd tell you to grab the big knob and move it up and down. You'd see this, the stage, you'd see it go up and down. Well, if you spin the fine focus, and you wouldn't see the stage move at all, because you use the coarse adjustment knob, the big one, to get it close and get it in focus at low power. And then when you switch to the next highest power, the medium power lens, if you touch that coarse adjustment, it's going to move it too far out of focus. And I'll explain why in just a moment. So you take your slide, and you put it in the slide holder here. I call it the little clippy thing. There's a little knob. You can see it kind of back here that you pull that little lever. This thing swings out. You put your slide up here into this corner, and then you just let go of it. And this holder here, this little lever comes in and will hold the slide in place. And then using your hours, the knobs hang out down under here. And this one there up here, then one of these knobs will move the slide back and forth in one direction and back and forth in the other direction, the other knob. And then you get your specimen so that it's right here in the middle of the stage where the light comes up. The microscopes have a light source and an on-off switch, of course, to turn it on and off. And the way this works is the light shines up through here, through this condenser. This condenser kind of focuses the light beam, narrows it, so it goes up through, this is the other end of the condenser here, and then the light goes through the specimen and up through the objective lens, and then there's a little mirror up here, and then the light goes through the ocular lens and into your eye. So the microscopes we have in our teaching labs, most teaching labs, the low power lens is 4, magnifies 4 times. The ocular lens magnifies 10 times. And so to figure out what total magnification is, you multiply the power of the ocular by the power of the objectives, and that gives you what magnification you're actually looking at. So for the objective, low power objective 4, the ocular lens at 10, the view at low power is enlarged 40 times. What that means is when the light comes through the out of the light source, through the condenser, through your specimen, into the objective lens, bounces off of that mirror, goes magnified another time through the ocular lens and hits your eye. That means that the image on the retina of your eye is 40 times bigger than the specimen actually is. And believe it or not, that doesn't sound like a lot, but that's enough to give you a view of a lot of different things. Sure, you might want to zoom in on some things, and then if you want to look at something like if we were doing this in the lab, we would be looking at slides of blood, some cells like I just showed you in the previous slide, sperm, because everybody will look at blood and sperm. I try to figure out what I can get people to look at. And blood cells and sperm cells are very, very small. So you have to use the high power, but it's also good because that gives you a little bit of exercise. If you've got microbiology in your future, then you're gonna quickly become an expert at using these microscopes. If you've already had microbiology, you know what I'm talking about. You use microscope obviously extensively in microbiology because, well, the cells are so small. All right, what other parts of the microscope we have here? So the ones that you're going to want to be responsible for. Let me hit those first. You want to know what the ocular lenses are and how much they magnify. You want to know where the objective lenses are. And you want to know the coarse and fine focus knobs, the name of the stage, and I think that's pretty much it there. What else we have here? As you can see, this little doohickey here is you unscrew it and you can rotate the head of the microscope. So if you want to show somebody sitting next to you, you want to show them what you're looking at. If you pick up that microscope and slide it across the bench, it's going to bounce, and your friend's not going to see anything. So what you do is you untwist this little knob here, and you can rotate the head, and your friend then can look in from over there without having to move the microscope. This might be the fine focus knob. What else we got here? I think that pretty much covers it. Microscopy is fun. You get to see all kinds of cool stuff. All right, and total magnification. Here's how you calculate it. You're going to want to be able to do this. The ocular lens, the total magnification is the ocular's only got one C. When am I going to learn how to spell? The magnification of the ocular times the magnification of the objective gives you total magnification. So as I just mentioned, the ocular is always at 10, and the low power lens is 4 power, so that gives us 40x or 40 magnification at low. So remember, then we do that first. We rotate the low power objective into place. Then we make sure our object is illuminated by the beam of light coming through the stage. You do that with your own eyeballs, not the microscope. And then you look in the ocular lens, and then you get your object in focus using the coarse focus knob, and maybe adjust it a little bit with the fine knob if you need to, and then you rotate the medium power lens into place. The medium power lens is 10 power, so 10 times 10, and you know, on the exam, you know, we're not going to have a calculator, but 4 times 10 and 10 times 10, I know you learned that in 3rd or 4th grade. So everybody's got their 10 times tables. Those are easy. 10 times 10 is 100 magnification. So that means at medium power, the image on the retina is 100 times bigger than it is in real life. When you use, if you need to, and you probably will, adjust it using the fine focus knob only. The fine focus knob only. If you bump the instrument or if you use the coarse focus knob by accident, it's not the end of the world. What do? You just go back to low power and start over. And I should, instead of saying if that happens, I should say when, because it happens to everybody. Everybody, all the time. Things happen while you're trying to go from one magnification to a higher magnification. Things get screwed up. You just go back to the last magnification you were using and start over. If you have to, go back to low power. It's no big deal. It happens to everybody all the time. So you just start with low power and you make your progression. And if something happens, the wheels come off. When that happens, you just go back to the last objective you used. And you'll get the hang of it. It probably sounds more complicated than it really is. In real life lab, in a classroom with 24 students, I probably can get 20 of them to be able to find an object like blood cells at high power, within about 20 minutes. So, but really is you just have to have your hands on. And I don't, it would be really cool if there was a simulator we could use. I may search see if I can find something like that. If you do, email it to me because that would be great. All right, so then at 10 power, you use the fine focus knob to get your specimen in fine focus. Then you rotate in the high power lens, which is on our microscopes 40x, multiplies 40 times. So 40 times 10, that's not too hard to do either, is it? So, that would result in a magnification of 400x. So this image on your eye would be 400 times bigger than it is in real life. That's pretty cool. Now, some microscopes in the A&P1 lab, they're not supposed to be in there. In the A&P2 lab, there are, and certainly in the microbiology lab, there would be another lens called the oil lens, which would magnify 100 times. So 100 times 10. I know you can do that. If you had 10 $100 bills, how much money would you have? See how fast you figured that out? It would be 1,000 power, and but it's called the oil lens, and I'll explain that from this picture. Alrighty, so you'll notice the lenses are of different lengths, with the low power lens being short and the high power lens being longer. And there's a couple of reasons for that. One, there's a distance away from each lens that's in perfect focus. In a low power lens, it's pretty far away. This is called the focal length, the length away from the lens where the perfect focus is. As you increase in magnification, the focal length gets shorter and shorter. The microscope is set up to be what's called parfocal. Boy, and I just can't even imagine trying to use a microscope that wasn't set up this way. The early microscopists, and I'll talk about microscopy and the history of that in the lecture when we get to the cell chapter. But so you'll notice that even though the focal lengths are different distances away from the lens, oh, where's my little circle thing there? It's set up so that where the area that's in focus is all the same place. And that's hugely important because each lens has a different depth of field. The depth of field is the slice of the universe that's in focus using that lens. The low power lens is very forgiving. So what would happen is you would put your specimen on a stage, and then you would rotate the coarse focus knob, and your specimen would rise, it would come up, and as it started to get toward the depth of field, you'd start to see something there, and then you'd be able to see it a little better. It would get crisper and crisper and crisper and crisper, and if you keep turning it, which you will, it's okay, it gets less and less and less and less, and all you do is just stop and go back until it's nice and crisp. Well, the depth of field is wide enough with a low power lens, makes it quite easy to use. But when you switch over to the medium lens, it's a little thinner. But when you switch over, since the microscopes are parfocal, the focal length away from the medium power lens, it should be right at about the same place. About. It's not exact. So you're going to need to use that fine focus knob to wiggle it around a little bit to get it in good focus. And that's not too hard to do with the medium power lens. But when you switch to the high power lens, the slice of life that's in focus. The depth of field is annoyingly thin, and it's not always real. It's so thin that it's not always in focus real well when you switch. Now, when I say you have to, when you get to this lens, you need to take great care with the fine focus knob. If you turn it just a tiny little bit, not even a tenth of a way around, it makes a big, huge difference. If you just grab that fine focus knob and turn it half of a distance, you're going to be way out here. You won't see anything, and you might be able to go backwards and then find it again, but you might not. What happens if that occurs? Well, you just go back to medium power, and if you messed it up so bad, you just go back to low power and you work your progression through. It's not too difficult. It's a skill that we want to have. So you're going to want to know what parfocal means. You're going to want to know what depth of field means. You're going to want to know the definition of focal length. You're going to want to know how to calculate total magnification, and then you're going to want to be able to recognize the oculars, the objectives, the stage, the different adjustment knobs. I don't know, the light source, that's easy enough, of the compound microscope. And that will get us started. So, coming back at you with cell anatomy next. So, arrivederci.